This Week in Virology, the podcast about viruses, the kind that make you sick. This Week in Virology, episode 18 for January 28th, 2009. From rainy and cold New York City, I'm Vincent Racaniello.
I'm Dick Despommier.
And from snowy and cold western Massachusetts, I'm Alan Dove.
Dick, are you in New York City also?
I am in New York City,
Yes,
Vince. I'm sitting right next to you.
Right across the desk from me. Welcome to TWiV, our weekly show about viruses. Today we have quite a show. And today Dick is getting ready to go to TED. So he'll be gone next week. But we'll post a link to TED so everyone can hear and watch him. They post the videos after the talks.
Right.
Now you're going to be really famous.
You will see someone shaking in their boots as they deliver their talk.
Although, you thought after TWiV it couldn't get any better.
This is true. And it probably won't either. Although I will try my best.
Good week for virology stories,
Yep.
I think. Oh, Alan, I had a question for you.
Sure.
Let's see, it came up in the car this morning. A couple of weeks ago, we discussed that you write a broad range of science, right?
Right.
If you could choose to do just one area, what would it be?
Ah.
Oh, well, it depends on how narrowly we define the area, but I would have to say probably molecular biology if I can define that as an area.
That's pretty broad because it covers everything.
Yeah, that's why I would choose it.
But you like that as opposed to conservation, global warming, etc.
Yeah. I mean, it's part of it is that there are fewer people doing molecular biology and covering that news, but also it's my interest, it's my background. There's a lot of cool science out there, certainly, but
It's interesting. You mind if I interrupt just for a moment? Because there was a wonderful interview with Ari Shapiro on NPR last week that leads right into this. You know what Ari Shapiro was trained as? He's one of the announcers on NPR, WNYC. It's an NPR station. They interviewed him and they said, Ari, could you tell us what you used to do? And he said, sure. I used to study killer whale behavior. Which is out of the blue comes, So everybody does more than one thing. And obviously, Alan does several things, not just two things. I know you do more than that because you have one of the most articulate ways of presenting verbiage that I have run across in a long time, Alan, and you must have some aspirations for acting as well. Tell me you don't.
I don't.
But you could. You have that kind of a voice.
Yeah, if something interesting came up in that area.
Dick, what do you do more than one thing? You do research here and you do vertical farming.
I do.
Right? I don't. I just do viruses.
No, that's not true, Vince.
I mean, I've tried to learn guitar, but
you're the heart and soul of TWiV. Come on, you're in communications now. No, you think that's trivial. Look at, he sits at a command desk that has all kinds of electronic gizmos, and we're bringing this to you through the kindness and inventiveness of Vince Racaniello.
Let's do viruses because I can tell people are
they're starting to fall asleep.
Impatient.
That's right. That's right.
All right. So this week, I am sure you all saw the stories about catch a cold and get fat, a common cold virus that may also cause obesity. Have you seen this, Alan?
Oh, yeah.
Oh, man, the Twitter is alive with this stuff. People saying, great, I get a cold and I'm going to get fat.
Or at least I have an explanation now.
So we will post links to the article. This was actually based on an article in the journal Obesity, which is a Nature group journal. And also, there's a history as well. And the idea here is that adenovirus type 36, so adenoviruses are rather large double-stranded DNA containing viruses. They're icosahedral, no membrane. Many serotypes, over 50, and they're associated with respiratory infections, enteric infections, a few of them with eye infections.
Why are they called adeno?
They're called adeno because they were first identified from
the adenoids.
Yeah, which are those things in the back of your throat, right? Yes.
Right.
In the 50s.
Right.
By Bob Chanek, I believe.
Uh-huh.
If I have to check that off the top of my head. I remember Bob Chanek discovering those. And the story here is that one of the serotypes, type 36, seems to be associated with a variety of phenotypes associated with gaining weight. So, for example, in the paper that just came out, in which the press is making a big deal over, they look at metabolic effects in cultured cells. They infect with this virus, and they see that it turns on an adipose program. The cells differentiate towards adipocytes. Previously, that in itself wouldn't be so remarkable, but there is a history. There have been some other papers with this virus. For example, there is a paper a while ago where they infect hamsters with this virus, and it looks like the infection affects processing of lipoproteins. Okay, they didn't look to see if they gained weight or not, but again, it interferes with the adipose program, if you will. Another study, they showed that this virus could be transmitted among chickens, which is not really a big deal because they didn't look at weight gain. But the one that is interesting is they inoculated, this is another paper a while ago, they inoculated chickens and mice with this virus, and they gained weight. They get increased adipose tissue, and you don't see this in animals inoculated with a control virus. Okay, and you can find viral DNA in adipose tissue of these animals. So that was another one.
Now, is the weight gain acute or chronic?
That's a good question.
And if you clear the virus, do you lose the weight?
These are very good questions, yeah.
Another question that
I think they didn't do that, Dick. That's a good question.
But even before that, you said the adenovirus was named because it infects the adenoids, or the lymphoid tissues in your throat. And yet it appears to be affecting the deposition of adipose tissues throughout the body. So are all cells in the body susceptible to the adenovirus series? Or
I think that the different serotypes have different tropisms.
Okay, so this one appears to have a very general tropism.
Well, this one can go to adipocytes, or adipocytes, I guess you say. Mm-hmm. And not all of them can, apparently, because in some of these studies, they've looked at other serotypes, and they don't have the same effects.
So does this not suggest a way of introducing a toxic agent
to the fat,
would selectively kill off adipocytes using an adeno-36 virus to deliver it?
Maybe, if in fact you can.
I mean, if they home in.
Well, I don't think it's specific for the fat.
I know I'm jumping the gun here, but
but the virus probably infects other tissues as well, so you'd have to have a way of getting it delivered to the fat.
We who are slightly overweight are looking for anything.
You overweight, Dick?
A little bit.
Another paper, they looked in the monkeys, rhesus and marmosets, and they inoculated 3 marmosets with AD36, and the monkeys have a 3-fold body weight gain.
Wow.
In 28 weeks.
That's a lot.
And again, I don't think they go beyond this observation period, Alan, or else we would know if it remains. So in other words, once the virus goes, does the weight go away, or is it permanent, or what? I don't know. Right. So, I mean, it looks to me that there is some involvement of this virus in adipogenesis. Right. I don't think we have the whole story. One fact we should also tell you is that this serotype is found in 15 to 17% of the adult population in the U.S. It's in 30% of the obese population, but only 11% of non-obese adults have this virus. It's associated with a greater body mass index. What I think needs to be done is, in cells, we know that this triggers a adipogenesis program. Find out the mechanism. Find the viral protein that does it.
Sure.
Use all the technology we have, cloning, gene silencing, transgenic animals. Look at the mechanism. And if you're really convinced that something is this, then go forward.
Right.
Because, if you say, this viral protein does this precisely, then you could take that protein, put it in animals, and show similar effects. And then if that all looked good, then you go back into people and do a big epi study.
That's right.
Right? So, and I think this is worth pursuing. I mean, I don't know these groups who have done this, and they publish in these obesity journals. They don't publish in the Journal of Virology or so forth. So I haven't seen it. I don't know them. But I think it's worth pursuing. And, a lot of people are poo-pooing this week, but I don't know. The data look okay to me.
What do they call obesity throughout the world? They call it an epidemic. So it's probably due to something that has emerged
on the other hand, this virus has been around for a very, very long time.
Well, that's the point. Maybe it's mutated.
Or we've got other factors that are contributing to obesity.
Yeah, like sort of, for instance, eating too much, or
Yes.
Well, yes, that's a great point, but it could be a predisposing factor.
Absolutely.
Right?
Yes.
And
yes.
but I mean, in the end, there are two components. We eat too much crap, right?
That's
we eat
right.
fast food, but there's
Yep.
also a genetic predisposition.
There is.
There are people who don't eat all that much and they get big.
Plus, there's a microbial story brewing out there also that I have to add here as a sort of another hypothesis to suggest that there are microbes that live in your intestinal tract, which have something to do with your appetite.
Right.
Yes.
And they only find these in obese people, not in non-obese people, and they can actually show, for instance, like germ-free animals, they don't have any fat deposition worthwhile. The moment they conventionalize them, the fat deposits jump remarkably. So microbes do play a role, at least in the way in which your body mass accumulates.
Yes. So that one person I know who studies that is Jeff Gordon from WashU.
Okay.
And that's very interesting. Do we understand what the flora in the gut is doing?
We don't even understand the flora in the gut, period.
There are a lot of flora beyond E. coli.
I would say there is.
We don't even have a census. Yeah.
That's correct. We don't have a census of the normal microbes that live in and on us, including the viruses.
Yeah.
Right.
How silly of us not to do this. I mean, we've got the genomics. We should just go ahead and do it.
Well, people are looking at microflora for sure. They're sequencing the whole gut, absolutely.
Right.
And that's where some of this work that you're talking about comes from.
Exactly right.
And so that's a great line of research. You're right. But this virus thing, I think it's worth looking at. So those suggestions I had, and then I have two more suggestions: eat less and exercise more.
Physicians say that all the time. That's their last words as you walk out the door, or as you waddle out of the door, I should say.
I guess we should just clarify that people are saying get a cold, get fat. The reason is that this, of course, adenoviruses are common respiratory pathogens. They're a cause of common colds.
So starve a cold, feed a fever, or what's that old adage?
Next, there are some interesting new inhibitors of influenza virus out there. And we have talked on this show about the three that we're using now, I think, right? Remantadine, oseltamivir, and Relenza. That's not the right name for that, right?
No, it's a trade name, I think.
Relenza is zanamivir.
Zanamivir, thank you. Thank you very much.
Thank you, Google.
That was fast.
Very fast.
Excellent. Anyway, we have H1N1 strains being resistant to oseltamivir this year in the U.S. Most of the H3N2s are resistant to remantadine. And so basically we need more.
Right.
We need a deeper pipeline because, with HIV we have 20 or more antivirals and we can make combinations of 3, right? When you get resistance to the 3, you just switch to another 3, and then basically you can live a full life with AIDS that way. But we don't have that with flu. So I did a little searching, and I found that there are a good number of new drugs in the pipeline for flu, which is good.
Yeah.
For example, there is one called CS8958. There's a catchy name. The companies give them these codes before I guess they decide they're going to be marketed. Then they give them a...
This is very common in the pharma industry. It comes out of...
Takes you right back to Salverson 606.
it comes out of the high throughput screening strategy.
That's right.
So they have tens of thousands or hundreds of thousands of compounds.
Exactly.
And they're all key coded this way.
And usually the name of the company, the first 2 letters are the name of the company, right?
Right.
So I guess CS, Daiichi Sankyo. So anyway, this CS8958 is another neuraminidase inhibitor. It's not a mimic of sialic acid like the current drugs are. It's simply a drug that was picked up in a screen for inhibition of neuraminidase. And apparently this drug, which actually is a modified version of a lead that they got originally, works on neuraminases of H1N1, H3N2, influenza B viruses. It protects mice from infection as well. And the cool thing is it also inhibits the neuraminidase of the current strains that are resistant to the current neuraminidase inhibitors. Okay, so we know we have resistance to Tamiflu, and they took those viruses, and they're still sensitive to this one. So that's not surprising because it's a different mechanism, but it's good because if you make a cocktail, then it's harder to get resistance.
Right.
So that's CS8958. I'll put a link to the article. That's pretty interesting. I also found that it's in phase 3 for adults, which means that the last phase of the clinical trial. And what they're doing here is interesting. They're trying to show that a single inhalation of this drug, so this would be inhaled, is as effective as 75 milligrams of oseltamivir given twice a day orally for 5 days.
Wow.
So that's what you need with Tamiflu for it to be effective. And they're saying that this one is going to be good with one dose. Well, we'll see, because I guess in mice the results were promising, and in phase 1 and phase 2 as well. So that's it's a new neuraminidase inhibitor.
And I love what that trial is called. It's a double-blind non-inferiority study.
What does that mean, Alan?
It's good to set modest goals. That's what I always aim for in the lab. Just a non-inferiority standard.
I have a general question I'd like to ask both of you, since you've had much more experience in virology than I have, and that is, what would happen to, let's say, a human being who lost all of the receptors for hemagglutinin?
He'd probably die.
What other normal function does the receptor for hemagglutinin play in the role of normal biology?
Sialic acid is a component of every cell on the surface, every glycoprotein, probably essential for cell communication, architecture, signaling.
So you're saying that
away,
the virus then takes advantage of something that we can't do without.
Absolutely.
In order to gain entrance.
Absolutely.
That's the point I was driving
at. As far
at.
as viruses can take advantage of, because as you know, they can't do anything.
No, of course not.
But yeah, it's an essential protein. It's an essential sugar. As far as we know, there's only one example of a deletion in a gene encoding a virus receptor that confers resistance to infection. You know what that might be, Dick?
No, I don't, Vince.
Alan?
I don't either.
It's the CCR5 deletion, and it's the co-receptor for HIV.
Oh, yes.
Certain strains of HIV.
Okay.
These people are resistant to infection with the viruses that bind to that receptor. We've talked about that before. So I don't know of any other example. So that protein is apparently not essential, but sialic acid, I would guess, would be. It's interesting that you say that, because there is another new inhibitor. It is called DAS-181. It's by a company called NextBio. It's also called Fludase. It's actually a sialidase. It's a recombinant protein with a sialidase from a microbe. And the idea is you would inhale it, and it would remove the sialic acid from your mucous membranes and prevent infection. So that's sort of, it's in line with what your question is, right?
Okay. So
that's DAS-181. That's in phase 1. So they're just putting it in some people, making sure everything is okay, right, safe. So that's a different step. That's blocking attachment. So it's different from neuraminidase.
Exactly.
There is another neuraminidase inhibitor, A322278, which is being developed by Abbott Laboratories. Again, it's active against a wide number of strains, and it protects mice against infection. It also is active against these neuraminidase mutants that are resistant to oseltamivir. And then, yes, Dick, you have a
do
question?
all of these inhibitors work against the active site of neuraminidase, or are they allosteric inhibitors or something else?
Well, the Tamiflu and the Relenzas are active site. They go right where the sialic acid would. They're analogs. I don't know what these do. These were just picked up in screens,
Because I was wondering about the...
So it could be allosteric or active site.
Yeah.
But Dick, the fact that they pick up that they're active against mutants that are resistant to the other drugs suggests that it's a slightly different site, right?
Right, exactly.
I'm sorry, go ahead, Dick.
No, I was just wondering how a virus could become resistant to a lookalike pocket molecule that inhibits the enzyme in its active site without altering the activity of its own neuraminidase.
Yeah, well, I think some of these... resistance wasn't so quick in coming with these new inhibitors, these new neuraminidase inhibitors, so that's part of the issue. But I think a lot of these resistance changes make the virus sick.
So when you line up all the active sites of all the neuraminidase escapees, how does that pocket look? Is it all the same, or is it lots of different amino acids, but as long as the charge is there?
Well, there are only a few changes that have been identified so far. It's not clear what they do in terms of the pocket. But, there very subtle differences can make a big difference in drug sensitivity. So it's really interesting. Yeah, you could imagine that even very similar inhibitors could have totally different resistance mechanisms as well.
Well, and on the virus side, this is a fairly constrained thing. You can't change the protein too much without losing your ability to bind.
Well, that's exactly right.
I mean, that's why these were originally developed, these sialic acid analogs. The idea being you can't change all that much because there are lots of other inhibitors for other viruses. It's easy to change. The RT inhibitors for HIV-1, it's a piece of cake to change because it's far away from the active site or it's in the active site, but it's easy to change one amino acid and get resistance. But I think in the end, the real answer is have a lot of antivirals, have more than a couple,
of course,
and use 3 at a time, and then you'll be okay.
Well, and it's encouraging, too. I saw you, in the notes, you listed one of these others, T705 from Toyama, which is totally different. It's a polymerase inhibitor.
Right.
And that's certainly a very encouraging route to take as well, because you want to attack the virus in multiple stages.
Absolutely, yeah. Having different targets is great. So not just all neuraminidase, right? So we have polymerase, we have HA, we have neuraminidase. I forgot to mention there's another neuraminidase inhibitor, paramivir, which is being developed by a company called BioCryst. So yeah, having different targets is good, and I'm sure you can find even more targets. These are easy ones because they're enzymatic.
Right.
So it's easy to set up an assay. In fact, the neuraminidase inhibitors, you just purify virus and put it in 96-well plates, and you can do a colorimetric neuraminidase assay and screen by robot for inhibitors through big chemical libraries. So it's easy. RNA polymerase is an enzyme. It's easy to do also. But there are certainly other targets. So the point of all this is that there's a lot of activity, and there should be more drugs on the market soon, because we need them if we're going to use them. Apparently, not everyone gets immunized. Only 20% of the U.S. is getting the flu vaccine.
Right.
So we got to deal with it. Plus, the other thing is, the last thing before we leave this, of course, if there's the next pandemic, we probably are going to need antivirals because we won't have time. It'll take a while to make a vaccine, and in the meantime, it would be good to have antivirals.
I would say.
And in that sense, these studies are great, but we're not sure if they would inhibit the next pandemic strain. In none of these papers did I see them testing an H5N1 avian strain. Of course, we don't know if that's going to be the next one or not. So,
right.
Okay. And there's also a great review that just came out this month from Frederick Hayden, who's a virologist at the University of Virginia. I saw him in Switzerland, and he's an antiviral guy. I'll put a link to that. It's got all this info in it. It's really good.
You mean he's an antivirologist?
Yeah, he doesn't like virologists. Next story is a brief one. A few weeks ago, we talked about Bill Gates giving money for polio immunization, and then before that, we talked about this expanded immunization campaign in Pakistan where they were going to immunize 3 million kids in 3 days. So here we go. Afghanistan. Snow insecurity hamper polio immunization.
Sure.
Over 650,000 children under 5 years old have missed the first nationwide immunization drive in 2009 because snow, troop movements, war. That's a lot of kids to not get immunized, right? And that just shows you can have money, you can have great vaccines, but if you can't immunize,
right?
Man, it's really sad. I don't want to beat a dead horse, but in light of what we talked about, it would be interesting to point out.
Sure.
It's not science.
Nope.
Okay, here you go, Dick. Here's a story for you. Virus could threaten Lake Champlain.
Uh-oh.
Viral hemorrhagic septicemia, also known as VHS. Isn't that a video cassette or is killing thousands of fish in the Great Lakes, and federal analysts say fish in Lake Champlain are at high risk of becoming infected. So this is a virus that does this, and it's bad for commercial and sport fishing, I guess, Dick, right?
Yeah, it's been in the past, it's been associated with salmon farming, particularly in Norway, where they farm salmon in the fjords, and there's hardly any movement of water as the result, and all the feces, which contains the virus, accumulates at the bottom of the fjord. And as the result of that, of course, it reaches a critical mass, and then you have this vicious cycle of the virus passing between the young fish and the old fish.
So when you have an outbreak in a farm like that, what do you do?
Do
you have to kill all the fish?
You bet. That's right.
And how do you sterilize the water, though, before you put the new fish back? You really can't, right?
You can't. So I think the fjords are lousy places for raising fish, and I think the salmon farmers in that area would agree. Whereas if you go up to Maine, where they have a lot of salmon farms also, you go up to the Bay of Fundy, You've got one of the strongest set of ocean currents in the world, and you don't have that problem there.
It washes the virus away.
Absolutely. There's no accumulation because you got these huge tides that change every 6 hours. So the West Coast has similar problems, though, in Puget Sound. Even though there are strong currents, they do have occasional outbreaks of this virus. And it's an economic thing, not just sport fishery. It's salmon on demand. We actually created this situation by almost extincting the Atlantic salmon.
So now we have to farm them, yeah.
So now we learned how to farm them, and in doing that, we may extinct not just the farm-raised fish, but the wild fish as well, too. So we have to find balance here somehow. And this points up the problem of monocultures. That's all I can say.
You know anything about this, Alan?
I saw this story. It's come up a couple of times. I didn't know about the Norwegian connection, but I know this virus has been endemic pretty much in European waters for a number of years.
Right.
I gather it spread to the Great Lakes through bilge water. Ships go over to freshwater areas in Europe, and then they transfer through the canals, and their bilge water contains
That's right.
either fish or just the virus.
Or zebra mussels.
Or zebra mussels, yes. This is how we get a lot of things. Of course, we've sent comb jellies back to Europe the same way.
That's
so
right.
It's a mutual exchange.
Plus economic disaster.
Yes. Oh, yes, that.
So this is a rhabdovirus. So it's a virus related to rabies virus and vesicular stomatitis virus. It's a little bullet-shaped enveloped virion with a single strand of negative sense RNA in it. And it infects fish. It causes hemorrhaging of organs, skin, and muscle. And they die. They can also pass the virus in the urine or the eggs or sperm or feces. And they say here that some of them get persistently infected. They're fine, and they swim around shedding virus continuously, so they're carriers of the
Vince,
virus.
I just want to say something here because I do a lot of fishing.
Yeah.
Fish don't urinate.
They don't urinate.
They do not. They urinate through their gills, so that's not really urine. I wouldn't go that far.
The waste is pulled away by the
All of
water
the
flow.
ammonia comes out of the gills.
But apparently, what you were saying, Dick, though, is that if the water exchanges often enough where they're being farmed, then that seems to help matters a bit.
Yeah, something like that old expression: the solution to pollution is dilution. So,
Yes.
I mean, in this case, it really is true. You have to reach a critical mass of hosts and parasites in order for it to get transmission. So
It's often transferred by bait fish. People catch live bait, and then they bring it to another area and contaminates it as well. They say the emerald shiner is a particularly popular bait fish in the Great Lakes region. You know what an emerald shiner is?
I do, actually.
Ever used it for bait?
I don't use bait. I'm a fly fisherman.
That's right.
It's a purist. So that's the other thing
I'm
you
not
do.
a
You
purist.
fish.
I just use flies. You can catch more fish with them.
So the only thing we can do about this is try and stop the cross-contamination, right? Because you can't vaccinate the fish. You're not going to make an antiviral. The bodies of water are too huge.
That's
And one last thought here.
Sure.
How do you how do the fish acquire this infection?
I think the oral route.
So the virus is on the bottom and it gets mixed up.
Yeah, it gets into the water column,
Fish ingest
and it works its way up through the water column until it gets to where the fish are again.
Is there? Should we worry about swimming in waters that contain these viruses?
Not for us.
You don't think so?
Not yet.
You don't think it could mutate, Dick?
Well, if you start breathing out of your gills, Vince, instead of urinating out of your, well, we won't go there.
Are
but
fish ancient creatures, Dick?
They are. They go back to the Devonian era.
You think they had viruses from then?
Had to.
So these are ancient viruses, probably.
I would believe that.
All right. I thought that was a good story to send you off to TED with a fish story, Dick. Alan, you're not a fisherman, are you?
I am occasionally.
Aha.
Not as into it quite as Dick is. But
Have you ever caught a fish with bulging eyes, bloated abdomens, bruised-looking reddish tints to the eyes, gills, skin, and fins?
no, I don't think I have.
Okay. Then you never saw a fish infected with VHSV, the VHS virus, the videotape virus.
Yep.
The videotape virus, right?
Okay. I thought I would mention next that last week, Alan, we did a nice little top stories in virology, important discoveries.
Right.
We mentioned tobacco mosaic virus several times, as it turns out that it's been important in virology history. Over at the Protein Data Bank, which is where they store all the coordinates for protein structures, so so that you can build your own structures on your computer. They have a protein of the month, and they have a nice picture of it. They generate a picture by a computer, and this month it's tobacco mosaic virus.
Outstanding.
And they wrote there, tobacco mosaic virus has been at the center of virus research since its discovery over 100 years ago. So I thought that was cool because we mentioned it was the first virus discovered, crystallized, assembled in vitro. So we'll put a link. This protein data bank is cool. And of course, if you have an iPhone, you could look at these structures on your iPhone with the program called Molecules. We talked about a couple of weeks ago.
So now you can infect your iPhone with tobacco mosaic virus.
Yeah, that's the way the logic goes, I think, right? And then you just erase it and reboot it, and you'll be okay. You don't need antivirals. Okay, Ebola in pigs and pig farmers in the Philippines. Turns out last year there was infection of pigs on Philippine pig farms with Ebola restin, the restin strain of Ebola. So let's back up just a bit. Restin is a strain that actually originated from the Philippines. And Dick, where do you think it was discovered?
In Reston, Virginia.
Right. So a bunch of monkeys got shipped from the Philippines to a primate facility in Virginia, and they got sick, and they were found to have this virus in them.
Right.
It infected some people, but nobody got sick. This is not a terribly pathogenic virus for people, as opposed to the African Ebola strains, which some of them are lethal, right?
Yep.
So this Ebola restin was found in some pigs last year. So they screened something like 6,000 pigs in the Philippines, and they found 4 seropositive or virus-positive animals. And so they were like, it's not very frequent. We're not going to worry too much. Well, last week in the Times, there was a report that a number of the pig farmers are seropositive, so they were apparently infected. So I thought this was interesting because, in Africa, the reservoir for these viruses, Ebola viruses, is believed to be the fruit bat or the flying fox. Now, Dick, are there flying foxes in the Philippines?
Absolutely. Oh, yeah, they're all over the place, all throughout Southeast Asia and even into Australia.
So can you provide a scenario for how Ebola restin in the Philippines got into pigs and pig farmers?
Sure, because it's probably the same story that you've got with Nipah virus in Malaysia, where the pig farmers decided to diversify and clear out a swath of jungle and plant mango trees. And so when the mangoes fruit and the mango fruit falls to the ground, the pigs would then go over and scavenge and eat the mangoes, and it would be sort of like a mutual situation where the pigs are being fed from the trees and the trees are being fed from nature, of course. And then when the pigs reach harvestable age, they're shipped off to market, and the trees continue to produce fruit. Well, the fruit bats like mangoes as well, but they don't like tree mangoes. They like the mangoes on the ground. So as soon as a mango falls to the ground, a fruit bat's right on it. We talked about this before in an earlier episode.
Sure.
I think something similar to that is going on here as well. The pig will come over and scare the fruit bat away, and the first thing the fruit bat does as soon as it's scared is to defecate, and then it flies away, as we would all do, of course, if we were scared by some animal 300 times larger than us, right? So you can imagine a scene out of Jurassic Park the same way. So here are these bat feces all over the fruit, and the pig, of course, doesn't seem to mind that, and ingests not just the fruit, but also the fruit bat droppings, and as a result, becomes infected.
Would a fruit bat go and sit on the pig food? If the pigs aren't around, would it go there?
Well,
be interested in that at all?
Pig food is whatever is available to eat.
What they put in the troughs, right?
Yeah, that's right. And it's not fruit.
So the
It's
bats
usually
are not going to
fruit.
go for that. So there would have to be some kind of a tree near the pigs. Okay.
That's correct. It could be bananas, though. It could be other fruits as well. Pigs will eat everything, but fruit bats will only eat fruit.
I think that's a likely scenario. We'll see.
Yeah. We should back up and mention also that of all the mammals on Earth, 20% appear to be bats.
20%?
Of the species.
Mm-hmm. Dick, is a camel a mammal?
A camel is a mammal.
Sorry, I just had to.
So if 20%, Dick, if 20% appear to be bats, how many actually are?
Well, it's hard to know because they mostly fly at night when we can't see them. Actually, Peter Daszak and I are friends, and he's in charge of a World Wildlife pathology group, and has done some wonderful work in tracing all of these emerging infections, many of them back to bat species, not just rabies.
There's a great article on bats in Microbe, which is an ASM publication. It's called Bats Prove to be Rich Reservoirs for Emerging Viruses. We'll put a link to that up. It's really nice.
Who wrote that?
Charlie Kalischer is the lead author.
Oh, nice.
And he is a guy out in Colorado who works on hantavirus. He traps mice and gets hantaviruses from them.
Great.
Now, in this article in the Times, they had a quote by a fellow from Galveston who said, "It's probably a rare event that pigs get infected. Perhaps so far it has been rare, but the rapid growth of farming is leading to more overlap between bat habitats and pig farms. It seems likely that farm animals will be taking more frequent dips in the zoonotic pool with dire consequences."
That's a great quote. That's terrific.
Yeah, maybe rare now. Another guy at CDC said that this virus, Ebola resistant, is very stable, not like flu or HIV.
Not like flu? I thought flu was stable, too.
He said there's no change of the virus when it travels from primates to humans.
I see what it is.
Well, that's the classic characteristic of a zoonotic infection, a dead-end zoonotic infection. So Can we recall what happened when West Nile went from a goose to a human? Did the sequence change, Dick?
It did. After a while, it did actually
After a while, but that first isolate was the same.
Now it was identical.
But now we know as it spread across the U.S., it's big-time change,
been modified, absolutely.
So I feel that that's a little bit of a misleading statement because we know all RNA viruses change tremendously, and it's just you give them the right environment and selective pressure, and you'll see the change.
Right.
But I'm sure Alan is sensitive to these journalistic representations, right? I mean, I don't mean to pick on them, but
no, it's absolutely true that my profession often gets it a little bit skewed, and you have to kind of feel for the science reporter who may or may not have a background in this particular field. It's therefore at the mercy of whoever answers the phone at the CDC is going to tell them what they're going to tell them.
Sure.
They've got to.
I think they all need to use Alan because he knows what he's talking about.
I would think so.
He was trained in a virus lab. I think if there's a virus issue, call Alan, okay, everybody?
Exactly.
That's
call
right.
Alan Dove.
That's right.
He's on
whatever you do, don't call me.
dovedocs.com.
That's right.
Okay, let's do a little email. I got an email from Greg, who's a publisher. He works for ASM Press. He digs up ideas for new books. What do you call that?
An acquisitions editor?
So he said ASM just brought TWiV to his attention, and he likes it. But he said, so in that episode that he listened to, we talked about the Cutter incident by Paul Offit, which is a book about this vaccine accident in the 50s. And he said he had a role in that book. So he had visited Offit a few years ago, and he told him that he was writing this book. So Greg brought it to his wife's attention, who was an editor at Norton, and she couldn't get it published. So then he showed it to his friend at Yale, and it turned out that Yale University Press did publish it, and that's the Cutter incident. So it's interesting that he has a little background there. And then he says his next book, which is the one about vaccines and autism, which got a lot of press recently, it was published by Columbia University Press, this latest book, and it's getting a lot of attention. So he said, at any rate, I wanted her, the publisher, to hear you plug the Cutter incident book at the end of the podcast. That was our book of the week. Chris writes, in the question about symmetry and consequences. So we had a question a few weeks ago. Someone said, is there any consequence of viral symmetry? We talked about it at length. I think you weren't here, Dick.
No, I wouldn't have.
One of the things I said is that when you have a filamentous virus, it can accommodate changes in genome. It can get bigger because it's more less constrained than an icosahedral shell.
Yeah.
So Chris says, I remember never being able to clone more than about 2 kb in M13. M13 is a filamentous bacteriophage. Because as the story went, the particles became unstable or fragile. So you really can't clone any length into the helical genomes. That's correct. It's not infinite, but it's more flexible than icosahedral capsids.
We misspoke.
Yeah, we misspoke. It shouldn't have been any length. It should have said more flexible. And then he says, we also said that envelope viruses, tend to be less stable, and that governs how they're transmitted and so forth. And he said, pox viruses are pretty stable in their envelope. You can freeze dry them, and that's how the vaccine is preserved. Yeah.
Yes.
I think pox are an exception. They have a very unusual structure. It is envelope, but beneath it they have this huge infrastructure of protein, so that may help the stability. But yeah, you're right. Thanks for pointing that out. Richard writes he finished listening to all TWiV past episodes. This guy's a medical student in the UK. I had an email from him before. He ordered a copy of Fever. Remember we talked about that, Dick?
Yep.
And he suggested another book of the week. You mentioned how interesting it would be to hear Jeremy Luban talk about the early days of the AIDS epidemic, how people reacted, what they thought was going on, how the scientific community tried to come to grips with it. This reminded me of a book I read called AIDS Doctors, Voices from the Epidemic and Oral History by Ronald Bayer and Gerald Oppenheimer.
Ronald Bayer, he's one of our faculty members.
Is that right?
He's right over here at our School of Public Health.
Well, I will have a look at that,
He's
and maybe it will be a future pick. But thanks
Absolutely.
for that suggestion.
You might even want to interview him.
Jeremy writes, and this is not Jeremy Luban, I have been listening to your podcast recently and found it interesting in the polio podcast that you said smallpox is the only virus that has been eradicated. I find this interesting since I have seen smallpox in the last 5 years. Specifically, when I was in Afghanistan, there was a boy that I came into contact with that had what appeared to be smallpox. Are there still areas of the world where smallpox virus is present? Now, smallpox was eradicated, and I doubt that you saw smallpox.
So camelpox, monkeypox, cowpox all can infect humans,
Yeah,
right?
they can cause a rash, sure, and other things can cause rashes, I doubt that it was really smallpox. But are there areas where smallpox virus is present? That's true, Alan, right?
There are areas where smallpox virus is present in some that we know about. I believe there's a freezer in Atlanta that contains some, and I think there's some in where is it, Sverdlovsk, somewhere in the former Soviet Union, and
somewhere else too.
Used to be some in England.
and somewhere else, and quite possibly in a few other places that we don't know about. So.
Yeah, so apparently the Soviet Union made lots of weaponized smallpox, but no one knows where it ended up. And so actually we have a book this week which relates to that. Everett had asked us a question a couple of weeks ago, and I asked Jeremy about it. And he was interested in these prostitutes in Africa that seem to be resistant to HIV and
They're
AIDS.
not called prostitutes, Vince. They're called sex workers.
Sorry. Anyway, he found an article about it. Apparently some of them are, in fact, quite resistant to infection. They're multiply infected and they don't get sick. And I don't know what the basis for that is. I don't think it's been studied. Here's an interesting one, gentlemen. It's our last email, second to last. Otar wrote a new approach to HIV treatment. He's basically saying that viral envelopes can be dissolved by short-chain fatty acids. And he said, why don't we add butyric acid to them? And it'll disrupt them and get rid of them, basically. And he did an experiment where he boiled butter and bicarbonate and got this cocktail, which he says dissolves stacked proteins. And if you could drink this, it would be great. And he says, there was a guy in London who had HIV, and then he got cured, and he's the only known person to have done this. And he made sandwiches, so maybe because he worked with a lot of butter fat, it was protected.
That reminds me, there was a story last week that I think we missed, and that was that there was a woman who caught rabies who actually survived, and she survived because they put her in a coma.
Oh,
and then
yes.
They gave her antivirals, and she survived it.
Really?
Yeah. She's the first person ever to have survived a full-blown case of rabies.
Wait, I thought there was one a couple of years ago where they did something very
they did.
similar.
They lowered his body temperature for a while, I think.
When Hillary Kaprowski was here, he mentioned a woman years ago that survived, but she was completely debilitated mentally.
Yes.
Maybe this is the second. So this was a recent occurrence?
Yeah, I saw
All right,
an
we'll look it
article
up
on
and do it next time while you're gone, Dick. Okay.
Thanks.
Anyway, so he wants to treat, HIV with butyric acid. I mean, so this is kind of kitchen chemistry stuff, but basically the levels of a detergent that you would need to disrupt virus in the blood would also disrupt your membranes, right?
One would think.
And I don't think this sandwich handler...
turn into a big giant lump of butter.
Which I think some of us are doing already.
That's right.
But that's because we have this weird adenovirus 36.
That's a different virus, yes.
Okay, our last email is from Pam, who's here in the city. I'm an ear surgeon who also does some herpes simplex bench work. Love your broadcasts, especially while looking at cells in the dark microscopy room for hours. Could you please do another in-depth episode in the future on Ebola? My nephew, 14 years old, has decided to become a virologist and also listens to your podcast.
Wow.
He has had this aspiration for over a year now. He really wants to work on Ebola. I'm trying to convince him to start with something a bit safer.
Yeah, that's right.
Well, certainly for the high school science
I would think.
I would do bacteriophages, yeah.
Exact.
Yes.
But I'd love to do a show on Ebola only and maybe get someone who works on it to talk with us. So that would be fun. Yeah, Ebola is pretty
Get
cool.
Dustin Hoffman to come over.
Yeah, I'm sure he would. Okay, that brings us to our picks of the week. This is a very exciting time when we highlight great science on the web and on paper made from trees. Science blog of the week is another one by this ID physician. We picked his podcast a couple of weeks ago called Persiflager's Infectious Disease Puscast. It's a really good, it's an ID guy who talks about his experiences. Actually, the podcast, he reviews the ID literature. But here, his blog is called Rubor Dolor Calor Tumor.
That's great.
And it's basically what happens to him on his consults. Interesting stories, commentaries on infectious diseases with rat holes by an inveterate persiflager. Dick, do you know what a persiflager
I have
is?
not a clue. I'm sure Ellen knows, though.
Ellen, do you know?
Well, I only know because it came up in the notes, and I looked
That's
it
not
up,
fair.
and then I saw that
That's
you
not
added
fair.
an annotation there. So yes,
Now,
that's
you've had to know that already. You
Someone who
can't-
banters.
You're just quoting the literature.
Someone who banters.
I'm a persiflager?
Yeah, you're a persiflager. So on this guy's website, he has a FAQ, frequently asked questions, and one of them is, what is a persiflager? And he writes, what, you don't have a dictionary? Can't Google?
He quipped.
So that's the kind of humor he has. This guy is great. I mean, he has really good stories, and
Nice.
I love reading them and learning too. So it's really podcast of the week. This is another interesting one. Organiz, Originz by Paul. A podcast.
Origins.
Sometimes you look at a word, you can't pronounce it. I'm sure it's happened to all of you, right?
Dyslexics of the world untie.
Oh, jeez. A podcast encompassing stories concerning just about anything and everything. There's information, theories, news, stories, conjecture, and ideas from history, geography, science, technology, language, medicine, archaeology, blah. You get the picture.
Right.
This guy has a really calm voice and really relaxing, and it's nice. I like it.
And a very narrow focus.
Right, exactly.
Yes. And finally, our science book of the week is called Biohazard by Ken Alabek. This is a story by a guy who used to run, apparently, the Soviet Union's biological weapons program. He defected, and he wrote a book about it. I read it. It's very enjoyable. I don't know how much of it's true. I've heard people say a lot of it isn't.
Apocryphal.
But it's fun to read. You can learn interesting things like if you want to weaponize a virus, you have to keep it lyophilized, and then you have 3 hours to get it into the warhead. So you have to be able to reconstitute it in 3 hours. And so they threw out a lot of viruses because they couldn't be reconstituted in 3 hours. If you add water too quickly, they fall apart. And he said filoviruses can't be weaponized, Ebola, because they're too fragile. So stuff like that you can learn. The smallpox trials that went wrong in the Soviet Union. Have you read this book, either of you guys?
No.
I have.
Yeah,
I have
you
not.
like it? Do you think it was...
Yeah, it was neat stuff, and as you say, it's pretty much impossible to know how much of it is true. Although there are parts of it that we do now know are true because of declassified documents that have come out and some of the stuff that was going on there. But it's a very interesting insight.
Do either of you know the origin of the biohazard symbol?
No.
You're too young.
No.
You're both too young.
I wish I were too young.
I'm not too young to know this because I was a graduate student at the University of Notre Dame in the Department of Microbiology when they sent around, I don't know who did this, I think CDC probably did it, a series of logos to represent biohazard. And we had to vote. And I actually got to vote on what I thought would be the best iconic figure to say, don't go in this room because there's some dangerous biological material inside. And it wasn't the one that they picked. I voted for another one, but actually it was interesting that they should have had to do this.
Does it mean anything, the logo, or is it just a logo?
Well, there are 4 intersecting,
Circular
curving
things.
lines,
Yeah.
which I don't know. I actually don't know. They never explained the figures, but they...
Yeah, it's always seemed kind of abstract
Yeah, me
to
too.
me.
They picked one, though, obviously, and now everybody's thoroughly familiar with it. And you just know origins. Speaking of origins, it started in 1964.
Now, I would have gone with Mr. Yuck myself.
Yeah, it's a circle, and then on top of that are 3 other circles, not complete circles, that intersect.
Right.
Sure.
And I guess it comes in different. It's supposed to be orange, right? Black
Yes.
on an
Yeah,
orange
it
background.
is
Right.
orange. That's right.
Well, it's in our heads now that this is biohazard, so
Yep.
that's pretty cool. Well, thank you for that tidbit of information, Dr. Despommier.
Well, I can contribute at some level.
You contribute at very big levels. All right, that's our show for today, everyone. Anyone have something they want to say before we sign off? Nope.
Well, except that I'll just miss you when I get down to that nice 75-degree weather.
I'm sure you'll have a good time. Good luck at TED.
Thank you.
All right.
Thank you.
I'm sure you'll do a great job. You're a great speaker and riveting.
Kind.
And steal that microphone, please. You can follow us at TWiV.tv. I'm PROFVRR on Twitter. Alan is Alan Dove, one word. Send us your questions and your desires and your criticisms. TWiV at TWiV.tv. You've been listening to This Week in Virology. We will be back next week. Alan and I, and perhaps someone else, whoever I can corner in the halls of biohazards here. And that should do it. Another TWiV is viral.